Myself, Sunil Kumar. I am doing PhD in Department of Pharmaceutical Chemistry, Faculty of Pharmacy. Today, I am going to discuss about what is meant by network pharmacology, why we require network pharmacology and all. These are my content. It means what is meant by network pharmacology, why we require network pharmacology, what are the key aspects of network pharmacology, followed by advantages, flowcharts, limitations, applications, along with some of the case studies. Coming to the network pharmacology. Network pharmacology, it is an interdisciplinary which integrated with pharmacology, system biology, and computational science to study the complex interaction between the drug and biological system and diseases. Wherein traditionally we will focusing on only single drug, single target, right? But in case of network pharmacology, we will focus on multiple drugs, multiple targets. That is the main advantages of network pharmacology. Why we require network pharmacology? As we know, limitation, that is single drug, single target approach, right? But in case of network pharmacology, we will focus on multiple drugs, multiple targets. That is the one of the advantages of network pharmacology. Again, biological complexity. It means we have to understand the deeper mechanism in some rare diseases like cancer, diabetic, neurodegenerative disorders and all. Advanced in technology, even though we have multiple target therapeutics, or even cost-effective drug discovery, personalized medicines, application in complex and rare diseases. Coming to the key aspect of network pharmacology, based on that is, first one is biological network and drug target interaction, multiple drug target interaction. Coming to biological network, which shows the relationship between gene, protein, metabolic, and its pathway in a system. For example, protein-protein interaction, protein-disease interaction, and metabolic pathway. Coming to the drug target interaction, where we will focusing on how drug interacts with multiple targets. It may be sometime it may affect on synergic effect, or maybe it may affect on antagonist also. Coming to multi-targeted drug discovery, we will focus on multiple drugs, multiple targets. For example, it is mainly important in multifactorial diseases like cancer and neurodegenerative disorders. Coming to the advantages of network pharmacology, that is comprehensive insight. It means we have to understand the deeper knowledge regarding the mechanism or pathophysiology of diseases. And drug repurposing, identify new therapies using existing drugs. Already whatever existing drugs is there, right? From that, we have to identify the new therapies for different diseases. Reduce the side effects. Allow the development of drugs with fewer off-target insights. It means one drug will bind another protein, except intent what we require, it will not bind. Other than that protein, it will bind. That we can reduce by network pharmacology. Again, at last, personalized medication. It means treatment which is based on individual genetic and network profiling. By surveying the person, we can survey the individual genetics and its network profiling. Based on that, we can go for treatment to personalized medicine. Coming to the flowchart. First, you have some of the extractions is there, or maybe polyherbal formulation. If you have any hypothesis, afterwards you have done any project, so it includes some extractions or polyherbal formulation, right? Afterwards, you have to go for analyzing the bioactive compound which is present in extractions or polyherbal formulations by using different techniques, is right? For example, GCMS or LCMS. Afterwards, you have to go for target identification for bioactive compound. It means what and all you will get bioactive compound, right? After analyzing GCMS or LCMS, for that you have to identify the target. It means our ligand on which target it will bind by using different software. That is one of the examples, that is Swiss target prediction. If you want to identify the target for disease, you have to use GeneCard or Malacard. These are the two websites used for identify the targets which is available for disease. Where in Swiss target prediction, it is available for targets which is depends on ligand. Afterwards, you will go for network constructing by using different software, that is string database and cytoscape, and we will go analyze of network construction by using different gene ontology analysis and Kaito encyclopedia gene and genome analysis. Coming to the limitation is data collection from databases, because more than one databases are available in website. Again, protein interaction also will occur. Again, multiple target can test other intermediates also. Again, standalone. It means no verified by experimental work and all. Coming to the application, drug discovery and development, identification of new compounds and new targets. Herbal medicinal research, explore the multiple component, multiple target nature of traditional medicines also, and disease mechanism understand. It means understanding the pathways and network involved in disease progression. This is one of the case studies. In this researcher, he has focused on inflammation along with squalene derivatives. He has chosen some of the squalene derivatives along with inflammation disease. Afterwards, he has extracted the targets by using different softwares. For inflammation, he has used DisGenet as well as GeneCard for identification of targets, for export of targets. Again, he has used Swiss target prediction also for extract the targets for that ligand. Again, he has interconnected for both the targets, that is ligand based as well as disease based. Afterwards, he has done for network pharmacology, and he has analyzed by network pharmacology, network construction by using different analysis techniques, that is KEGZ and GO analysis, and he has validation by using molecular docking studies. Coming to the case study 2, here researcher has focusing on myocardial infection of Glaucozebra bania decoction. It is an extraction of plant where he extracts the compounds from TCMSP. It means traditional China medicine for system pharmacology, and he has identified the targets for myocardial infection by using GeneCard, Omic, Pharma GKB, Drug Bank. These are the websites to identify the targets available for myocardial infection. Afterwards, he has done network pharmacology construction by using different software like cytoscope. He has used again string as he has used. Again, he has analyzed by using different analysis technique, that is gene ontology analysis and Kaito encyclopedia, gene and genomic enrich analysis. Afterwards, he has done validation by using molecular docking and molecular dynamic study. These are the references for your reference. After doing GCMS and LCMS, I got the some of the compounds, that is bioactive compounds for Alzheimer's disease. For example, I extracted one plant by different methods. I will go for bioactive constituent analysis. Afterwards, I got some of the compounds. For example, epigenid. I got the drug, that is epigenid. Afterwards, I will use the tool, that is Swiss target prediction. See, if you search Swiss target prediction, this is the outer space of that Swiss target prediction. Just paste your smile of your target. That is epigenin, right? If you search in PubChem, you will see the smiles of that. Open PubChem, search epigenin. Afterwards, you have to open that outer face. You can see here smile of that structure. Here you can see it, right? Just copy. Here you have to paste. That's why you can see the structure of that smile. It means epigenin. Now I inserted the epigenin drug. Predict targets. Your job was submitted. Just wait for 1 minute. Here you can see the target. It means your drug will bind to this target. It means whatever drug it is a right bioactive compound, that is epigenin. That drug will go and binds to this target. To identify this one, we have to use with target prediction. Afterwards, you have to download it this file in Excel format. You can see the options here. Just save that file in Excel format. After getting downloaded, you can see that file. You can see that target, that is NADPH oxidase 4, that is the common name NOx and Uniprot ID, Campbell ID, everything you can see in this file. Okay. Next one, that is gene card. Before opening, you have to log in with your Gmail ID. Okay. If you open that gene card, this is the outer face of that gene card. Just you have to search your disease name. Like for example, Alzheimer, right? Just type Alzheimer. You can see 15,557 targets are available for Alzheimer's disease. You can see it right here. Just download that Excel format. Here you can see that export to Excel. This one is that I will tell it in network pharmacology. That is only. Yeah. That only I will tell. By drawing the network, we can choose top 10. You can see it here, two folders. That is with target prediction, which is depending on ligand, and gene card, which is depending on disease, right? Now we have to merge two. It means, have you studied right in statistics? A union B. It means in both the files, we have to extract the common targets, like A union B. Like what is the Swiss target prediction and gene card containing similar target. For that, you have to use interact when as a web server. For that tool, interactive when. Interactive when diagram. You can see this one. It means you have to meet whatever ligand targets is there, ligand targets and disease target. In that, what is the similar targets available? To extract that one, you have to use interactive when diagram. Here, left, right side, you can see the options. Now, two targets. One is ligand and second one is disease is there, right? That's why you have to select two. Just select two. Set A containing ligand target. Set B containing disease target. right? Now you can see, right now it is ligand target as 0. Again, disease target as 0. Just you have to copy the targets. Already you exported, right? Gene called and Swift target prediction. Just copy the common name and paste that file. I will show you. Open Swift target prediction file. You can see that, right? Common name. Just copy this common name. Copy, paste here. Now you can see that. Now ligand target 100 is the right 100. Here also 100 is there. Right now I didn't insert disease target, right? Now I will insert ligand disease target. You can see gene card, open that file. Here you can see gene symbols, that is common ID for target. Totally it is having 15,575 targets is there. Copy, paste it. Now you can see this is the ligand target, that is 100 in nature, and this is the disease target, that is 15,557 is there. But this is the 90. It means both having 90 similar targets. It means ligand target containing 90 has similar to disease target also. It means that is the both the target having common targets. That is, just click on that 90. You can see that list. These are the common target for ligand as well as disease. Just copy this one and paste in Excel. Type it as hub genes. We will call as hub genes. It means common targets for ligand target as well as disease target. Just paste here. You can see it 19 right now. Just click on that. You can see this. Just copy this one and paste in Excel. Type it as hub genes. Here, write it as hub genes. We will call as hub genes. It means common target for ligand target as well as disease target. Totally, it is having 90. Afterwards, we will go further network construction. For that, we have to, before going to, for that, it is having two tools is there. One is string database and second one is cytoscape. It is a app. Okay. String database and cytoscape. Two tools will be there, that is cytoscape and string functional database. If you search string database, you can see that string functional protein associate network. Just click on that one. This is the outer phase. For that string, just click on multiple proteins. Right now we have 90 types of targets, right? That's why I kept as multiple proteins. Just here paste your hub gene. Whatever you extracted 90, right? That you have to paste here, list of name. Just copy, paste here. Now we paste it among common genes or common proteins. Now we will go for working on Homo sapiens, right? That's why we have to select Homo sapiens. Sometimes if you are working on bacteria, different criteria is there. You have to select according to that your work. Right now I am going for Homo sapiens. Now search. Give continue. You can see it, right? This is called network pharmacology. It is like single root containing different nodes. Path. For example, this is a protein. Like for example, this is SRC. You can say thread-like structure, right? It means different path. It means it is interconnected to other protein also. If you suppress this protein, all other proteins also will get suppressed. We will call it as node. That we will call it as node. Downside, you can see that here viewer option is there. Here analysis option is there. Here you can see that different colors. It means red color means single protein which is having different connection. It means it is having multiple connection. Fully red means it is having most top target. Afterwards, you have to go for cluster. Here you have to select K-mean cluster and apply. It means we will get only top target. Just click on that apply button. Okay. Here you can see that how to clustering that. Here you have to select K-mean cluster. It means based on top proteins is that, right? Based on that only it will segregate the top 10 or top target. That is why you have to click on K-mean cluster and apply. You will see the only top target. You will see the only red mark. If you different clustering analysis also is there, you can choose K-mean cluster means it will give based on only centered. It means only top. You can see downside. You can see that one. First cluster containing 72. It means that single protein containing 72 genes connected. It is connected with 72. If you suppress any one, in that 72 will also be suppressed. Again, different analysis technique having different manual. MCL cluster, it is based on this one, stochastic flow. Again, some randomized also is there. Again, from graphical also it is there. Now, just apply. Afterwards, you have to export this network in Cytoscape. For that, you have to click on export. You can see the option that is send network to Cytoscape. Just click on export. Afterwards, click on send network to Cytoscape. Afterwards, it will directly, this network, it is going to Cytoscape app. You can see here. Whatever we done right in a string database. Here you can see that one. Now you require top 10 genes, right? For that, we have to use this software that is Cytoscape. For that, you have to add one more application that is CytoHub. This is the one of the extension for Cytoscape. You have to download Cytoscape along with CytoHubba. Hubba. Yeah, the free only. This is the free tool. Just click CytoHubba. You have to download Cytoscape along with CytoHubba. That is the extension of that to determine the top 10 compound, top 10 genes. CYTOHUBBA. CytoHubba. HUBBA. Just click on CytoHubba. That is the extension of cytosome. Here you can see the calculate. Click on that calculate. Here top. How much you require? Top 10 genes you require? Type 10. If you need 20 targets, type 20. It means here number of targets will be there, right? Out of that, it will give top 10. Right now, I am clicking as top 10. Afterwards, submit. You can see it. These are the top 10 targets. We got 90 targets right before, but in that we will shortlist it and we will extract the top 10 targets out of 90. You can see that names also. If you want arrange, you can extract this picture also. If you click any one, you can see that display name. This is the target, ESR1. If you click change, you can change the name also. See that. This is EGFR. This is MMF9. This is MET. This is PARP1. You can export this image also. Here you can see that option save. Save as image format. You may adjust the quantity, quality and all. Okay. Now you can see. You can keep it in your publication. These are the top 10 genes. Again, if you want to list also, it will give in Excel format also. Here you can see the tabular column, right? Here save current rank in text file. You can see in CSV file or text file, anything is fine. Right? Result of network pharmacology. Save. You can see that one. Along with the string IDs. If you type string IDs, you will get the name of that target. This is the top 10 targets. You have to work on this top 10. Whatever you will get the bioactive compounds, right? After GCMS or LCMS, afterwards you have to, all the compounds you have to download by SDF format. Afterwards, you have to download these top 10 compounds. Again, you have to validate by different mechanisms, that is molecular talking or molecular dynamic studies. This is the network pharmacology. Yeah. Yeah, yes, ma'am. Yeah. We have to download in SDF format. That they will teach next class. Talking, dynamics and all. Anything doubt is there? Ma'am, there's no any preliminary study. Just download Cytoscape along with CytoHubba. Enough. Yeah, you can apply. You can apply every small molecule, even large molecule, because we'll go for extractions only. Extraction means it is a plant extraction. It is containing small molecule from small molecule to larger molecules.
If we are synthesizing new molecule,
that yeah, that also you can apply. Like if you draw that compound in ChemDraw, you just copy this mice and you will get the target.
Thank you.
Hey, Sai.
So can you please go to the cytoscape software? Can
Which
you
one?
please go to cytoscape software? Yeah. So in this, while taking a top 10 genes, you are chosen a MCC as a method.
Yes, sir.
So why not any other methods like MNC or degree?
Yeah, you can choose MCC as a method and different method also. You can justify that also.
Which
Right
one
now,
is
I
the
am
most
using
reliable one?
MCC only, but I have to study that one. I will rectify in best.
I am asking which one is the most reliable one?
If you choose MCC method, it will give more accurate as per literature release.
As which is MCC a reliable one? Thank you.
Right now, I am running. If I know miss, I will contact you with email. If you want to do multiple compounds like that,
So when we are going for polyherbal extraction in LCMS also, we'll have many compounds which are core. So in that case, how can we run a network for all the compounds together, particular target, determining of targets?
Yes. Actually, right now, due to shortage of time, I chosen only one drug. Because of that, we have to choose multiple drugs, take all the common symbols. That is genes, whatever we will get different targets, different right. In that Venn diagram is there, right? There is an option 2, 3, 4, 6. That we have to select how many compounds we have. That much we have to select and we have to paste in that.
Then we have to finalize
Yeah.
the genes which
Yeah.
is coming.
Yeah, you can do it. Maximum 30, 40, 50. How many compounds you have? That much compounds you can do it.
If the number of compounds and the structure will be different, right? They won't have the similar kind of structure. When we run at a time, they will have the common receptors,
Now, once
targets.
going to the synthesis, you have similar structure and derivatives, right? That is the one criteria. If you want to extract, in that one compound may be act, another compound may be not act. But all the compounds we have to test.
Can I just go back to the string database? Yeah, in this, can you explain a bit more about analysis part there?
Yes. Clustering, right, sir? Clustering, how to make that one.
Yeah, I just want to know what and all options can you use in that analysis?
Here you can see no cluster means it will give single, only limited quantity. It means there is no accuracy. It means there is no top 10, there is no interlinked with other proteins. That is that. If we select K-mean cluster, it will give based on center. It means top compounds is there, which is having higher network connection.
You are explaining about the cluster.
Yes.
I'm asking about the analysis part.
Analyze.
Yeah.
Don't know. Analyze. Sir, doubt.
Okay.
I don't know about analysis. What is that?
Yeah. Can you
Repeat
just,
the question.
can you just explain, can you give some details about what and all options we can use in this analysis part?
So here, the basic outline is what are the various properties we have, right? From this in general will be taking the came in only. After that, in cytoscape, as due to the shortage of time, we couldn't able to explain more. As you raised, right, only why you have done this method. There one more method is called as edges and nodes. We'll be trying to connect all the edges and nodes, and we'll try to get the data. So this here will be using as a primary tools, as a resources only. Here we are not going to do much analysis, and we are not exploring with those things because we are not a bioinformatician, we are a pharmacoinformatician. We need a right target only. If in case I fail to work on or completely if I'm working as a bioinformatician in the sense, then I have to go deep into that particular gene and which gene is upregulated down, then I can able to do it. But as a part of drug discovery, I'm not much into the bioinformatics, only into the pharmacoinformatics base. We are working on that. And that too, max next he showed right cytoscape will be importing. That after importing, we'll be calculating the edges and nodes. Among the 10 we have showed, from the 10 also top one you can predict based on their scores. Example, I'll be, 5 people are there in the group in the sense, among the five are the top. But within the 5, who is the topmost? To predict that, who is having more interaction? Okay, I'm the person, and with me 4 are there. I'll be interacting with him in the sense. So if you convey something towards me, I can convey to them easily. That's a concept. So if you suppress me, they'll be suppressed. So the major target, the pathway will be involved. So that will be working much more into the cytoscape only. There's the here showed up the cake pathway and all, right? That cake pathway will be further after finding this map, that will go into the cake pathway. Then from there we'll be adding the edges and nodes and all, which is the possible, the best, the highest number which is having highest edges and nodes. So that will be considered the topmost protein. From there we'll be working under. So here and then ma'am have asked whether only one compound or 2 compounds. As I show here, showed the target prediction, right? So once you've done the target prediction, you'll be getting such kind of a list of targets. So that targets, again, for the second compound, third compound, you can make n number of compounds. You can make a single Excel sheet only. That sheet, it won't matter. Because as a plant product, we, what do we call it? As a science, we in a cognitive way, we'll be saying, I have taken this extract and it's having polyphenols, more antioxidant will be same story. But what type of compounds it is present? We have to identify what mechanism. For that, this network formula you are working out 4 compounds, 5 compounds, most probably to which target they will be focusing on. That can be done over here. That's the thing.
Sure, thank you.
Any other questions?
Here we have some 2 questions in online. Like if we want network pharmacology for 2 bioactive, then what should be the procedure in that?
Same thing we have discussed.
The same.
Just copy that smile, first one, and you have to go in a switch target prediction. Just download that target of that ligand. Afterwards, again, you have to go for switch target prediction. Second smile, you have to paste and you have to export that file in Excel format. Afterwards, you have to merge 2 files. That all the targets from first target as well as second target you have to take and you have to insert in interactive Venn diagram.
One question.
Any other queries? Tech team, any online questions?
Can you go to that string file, string Excel sheet?
Excel 6, sir.
Excel. No, the first one, first string file. That
Yeah.
probability, no. Probability file.
Objects, sir.
That not that one. Not that one. No, sorry. Sys
Target prediction. That was in open.
Okay, so here actually in the case of epigenin, the probability will be 1. Probability is 1. This much of targets. Yes or no? That means the compound is highly active in these targets.
Yeah.
Okay. So suppose I'm getting n number of targets with lesser probability. What I have to do?
N number of compounds with?
N number of targets with lesser probability.
Then that compound may not be potent. As I said, right, in a plant, it may be 40 to 50 active constant will be present.
I'm not asking about the plant. Synthesized or design.
Yes, the same way, again, I'll say, because some functional group without some structure will be designing, where we may not have the functional group also. That time it won't go and bind, right? Same way, if you are not going to get that thing, this compound may not be going to work out. I mean, the design structure, we may have that idea, like this can able to go and work on those things and all, gene cut method and all. As an example, I'll say, if you have designed a compound of a pyrazole ring for a particular disease, and you are asking that if I upload to this, and if I'm not getting that target, that's the right.
Yeah.
So
No, I uploaded the target, uploaded the smiles, but I got the target. But the thing is, actually, I didn't get 1. 1 is that is for maximum probability.
yeah,
That means that target is okay with that.
best to move on. Okay, right.
But
Yeah.
I didn't get 1. Okay, that is my question. If I am not getting that 1,
then we can't able to go with further with your compound. Design compound, we have to modify. Any functional group you have to change and you can try. That's the option. Rather than that, only as of now, 6 target protection is available to find out with the smileys. For the genes and all, as you discussed in Mala card will be there, gene card will be there, multiple options are there. But time being, we have that only one database where it can be able to work on. So if it's not giving a score, we can't able to move further.
If it is not any plant product, if it is any monoclonal antibody or any kind of this structure, it is applicable in this software.
Monoclonal antibody.
No, ma'am. So 2 more online questions there. We'll
Here
answer
are
them.
few online questions. From Shilpi Chauhan. Sir, should we choose 4 pair in interactive wean for 2 bioactive?
No, no. Only one we can do at a time. We will make a video and we will share the online participant.
One more question.
Someone have asked about the cake pathway and geo analysis for a demo. We'll make a short video and we'll be sharing to the same IDs, same mail IDs. We'll be sharing how to work, how to choose a cake ID and how to go with the gene ontology and that.
Any other questions from participants?